free water, et cetera, stuff like that, in their workflow so that when you receive those samples from the pathology core that they're good for, molecular assays versus, something that's less sensitive, that they might be more used to working with. And I think for something like Visium, where you have to place those sections into small fields, or Visium version 1, I guess, there could be a lot of training for that as well. So, it's been a lot of back and forth between the cores, and then I have also kind of learned from the histology core myself to kind of learn how to section. But even that, I think I think Carla mentioned that genomics course taking on that kind of work isn't necessarily best, and I would agree, even though it's myself doing this. I think that the quality of my work is definitely not as good as some, a histology technician who's been doing that every day for years and years. So I think finding those connections can be really important instead of trying to do it all in-house, because it's a large undertaking, even though it is tempting to make it easier on the collaboration to try and do everything yourself, but I don't think that necessarily is the best way.
Yeah, 100% agree on that. And I mentioned the part of ownership. These assays are very long, and they start actually when the sample is being collected, even before somebody thinking about doing spatial. And when the sample is being archived, cut, and each tissue has its own challenges. And as you move forward to the processing, imaging, sequencing, if something through this chain breaks, you will not get good results. So having either through a collaboration or internalized, but a good control over the process and people being invested that the final product is good. We have found that when we collaborated with cores, showing them the results and involving them in what's the outcome of the process and how that sectioning looked like. And if there is a bubble, look how Visium looks like. And if that also helped our histotech being invested. Anuja?
Yeah, so my name is Anuja. I oversee a sequencing group at Stowers Institute. We don't work with human samples. The most normal organism we work with is mouse, and we work with a lot of non-standard organisms. I say that because some of these approaches don't really work unless you're working with human and mouse, right? So we started doing slide-seq, I would say, about 3-4 years ago. And then since Curio came into the market, we have switched to doing Curio. We work very closely with our histology core as well as our imaging and bioinformatics course. Anytime somebody wants to do a spatial project, we would meet as a group with the lab. And that's how we get started. And we have workflows documented that we follow. We know when a project is coming, when histology is doing the sectioning, when we have to be ready for doing the wet lab work, all of it. Recently, we ran into an issue that I want to bring up here. We were buying all the Curio kits and giving the slides to histology team to process. And we were charging back the labs. The histology team didn't realize how expensive those slides were. They just started doing sectioning, and the sectioning didn't go well. And they went through 4 1-centimeter slides and blew 10,000 K in 5 minutes. Since then, I make them buy the slides, and they were aware like how expensive these were at some point. They just forgot. But Curio has been good about dividing this slides from the reagent kits and sending them separately to us. So some of these things you live and learn, right? I mentioned we work with all kinds of different organisms, and that has been a challenge for us when we are looking at these in situ probe-based instruments. So we have generated data from NanoString, Vision with Iannis's group, Resolve, and we are about to generate some data using the Cinium with Washu. And I think right now we are leaning more towards bringing a Cinium in-house. For a while, we were talking about bringing the Vision But just because we are so familiar with 10x, it seemed like we might go the Cinium route.
Anuja, I have a question on that actually. So you are using a lot of this custom probes, especially like Cosmex kind of platform, which those custom probes are very expensive, like $350
Yes.
the gene. So which means like, and then they claim that it's for 192 samples. So how you actually really deal with it? Like you just pass on to that investigator, or you buy custom probe, keep it with you, and then divide it among the users? What's the strategy there?
Yeah, so Bonnie, that's a really good question. That's one of the reasons we are not going with Cosmix. We kind of liked the data and how their software seems really nice, but we decided not to go with Cosmix. With NanoString Cosmix because the kit sizes are so large. Vision and Cinium have smaller kit sizes, which are more appropriate for the kind of work we are doing. We do discovery work. So we won't be using the same panel like 100 times. We might design a panel, run it on for half a dozen samples, and that'll be it. Right now we are going to be charging back the labs for the whole design.
Yeah, just a quick comment to piggyback on Anuja's point. So before Xenium offered a full custom, Xenium, Cosmix, and Meriscope were three vastly different products, even though the underlying technology is very similar. You had one that is a very large assay, but it's very difficult to add to it. You have another one that is fully custom, but you don't have modules or anything pre-built that you can use. And you had another module-based assay. Now that Xenium is adding that custom, then it's a bit closer to Vision. However, we are doing mostly custom since we have Meriscope. We have found that our clients, for instance, they cannot design a 500 plex assay that makes sense. So we now have a computational biology team that is designing, because most of our clients came and said, we want to have 500 plex. And it's okay, do you have any genes you're thinking about? Yes, these 30. And we want you to add 470. So that needs single cell experience, that needs domain experience, and we have a team now dedicated to do that. So custom comes with a benefit, which is you can design whatever you want, but that puts more pressure on designing a good panel. Douglas?
Yes, thanks. Hi, I'm Doug Todges. I'm at the University of Vermont College of Medicine, and we're pretty new into the spatial omics game. We just created a Center for Biomedical Shared Resources. We moved into the first floor of a new building, and we brought together in one location genomics, proteomics, flow cytometry, bioinformatics, and microscopy imaging. And so we purchased a NanoString GeoMix. We have it housed in the imaging center, but the imaging center is directly connected to genomics. So we do and we also have histology lab in the microscopy imaging. So we do all the histology, the sectioning, the preparation. We then do the staining, the NanoString GeoMix work, and then we just take the plates through the door to the genomics lab, and they do the sequencing and then the bioinformatics. So we're early on, but what we found is, as others have said, the communication is absolutely critical. So we as core directors talk to each other daily, but we set up consultations with clients who have a project that they think might be amenable to spatial omics, and we go through all the details. We set up timelines, we communicate to make sure that when we know the sections will be prepared when the kits come in and that the sequencer is ready. So it's a lot of coordination of the cores, but so far again, we're early on, but it seems to be working pretty well.
Thank you, Douglas. And we have 30 seconds, Prashant. First of all, thank you everybody for this. I think we should meet more often, especially for spatial. I think there's so many things I would like to ask and get help. But so Prashant,
30 seconds. So thank you. Quick question about the custom panel. So I was talking to the people from Resolve Biosciences. I don't know whether you guys have anybody using that platform, but seems like for them, no matter which set of custom genes you choose, that doesn't change the pricing. I don't know whether that's true or not. Have anybody in the in this group is using Resolve?
So I think
For
for any,
Spatial?
for any custom, the number is flat regardless of what gene you want.
Yes.
Yeah, all custom are like that.
Okay.
Just the number dictates pricing.
Yeah, but I think what they said, like, even if you do like 4 or 5 samples, you don't have to buy, custom babes for like, hundreds and thousands of samples. But you can run, so 4, 8, 10 samples, and that doesn't change pricing too much or at all.
Closing, but we need to ask.
20 minutes is not enough, man.
Yeah,
Never enough.
Not enough time.
yeah, no, the discussion's just going, and I think this is fantastic. So we have, I'm gonna we can do the recaps that we've done in the rooms, or we can go back and do